In this quarantine, I have been able to rewatch some movies. One of those movies I rewatched was Jurassic Park, and while learning about biology, one scene in particular caught my attention.
What? What? Oh, Mr. DNA, where did you come from?
From your blood. Just one drop of your blood contains billions of strands of DNA, the building blocks of life. A DNA strand, like me, is a blueprint for building a living thing. And sometimes animals that went extinct millions of years ago, like dinosaurs, left their blueprints behind for us to find. We just had to know where to look. 100 million years ago, there were mosquitoes, just like today. And just like today, they fed on the blood of animals, even dinosaurs. Sometimes, after biting a dinosaur, the mosquito would land on the branch of a tree and get stuck in the sap. After a long time, the tree sap would get hard and become fossilized, just like a dinosaur bone, preserving the mosquito inside. This fossilized tree sap, which we call amber, waited for millions of years with the mosquito inside until Jurassic Park scientists came along. Using sophisticated techniques, they extract the preserved blood from the mosquito and, bingo, dino DNA.
With this being said, in order to understand DNA better and hopefully one day recreate dino DNA, today we're going to figure out how DNA and RNA work. First, we need to start by understanding that DNA is a double helix. It has two strands that twist around each other, and each strand is made of single units called nucleotides. It also has a sugar phosphate background and bases that join the two strands by hydrogen bonds. These bases are cytosine, guanine, adenine, and thymine. Complementary bases pairing is a key idea in genetics. C pairs with G, T with A, and each strand of DNA can be millions of base pairs in length and is coiled up to make chromosomes. Nucleic acids, which are DNA and RNA, are very large molecules that are constructed by linking together nucleotides to form a polymer. The nucleotide is made of a phosphate group that's acid and negatively charged, a covalent bonds that hold all the nucleotides that are very strong, a sugar made up of 5 carbon atoms making it pentose, deoxyribose for DNA and ribose for RNA, and a nitrogenous base that contains nitrogen where A, T, G, and C is located. For both DNA and RNA, the composition is adenine, guanine, thymine, and cytosine, with only thymine changing for RNA to uracil. The sequence of bases makes up the genetic code. Nucleotides are linked into single strand via condensation reaction bonds are formed between the phosphate of one nucleotide and the pentose sugar of the next. This results in a phosphodiester bond between the two nucleotides and the formation of a water molecule. Successive condensation reactions between nucleotides results in the formation of a long single strand. Among the differences that we can find between DNA and RNA, one is the number of strands, as DNA is single-stranded and often but not always in linear shape, and RNA is two antiparallel, complementary strands from a double helix. DNA in and of itself is a double strand of polynucleotides. The sugar phosphate backbone is on the outside and the bases are on the inside. The strand is held together by hydrogen bonds between the bases, and A only pairs with T and G only pairs with C. This is called complementary base pairing and is super important. The two strands run in opposite direction and this is called antiparallel. This is why DNA twists into a double helix held by more hydrogen bonds. The double helix is also maintained by hydrogen bonds that hold adjacent sections together. Hydrogen bonds also hold complementary base pairs together. Complementary base pairings ensures that mistakes are not made when copying or transcribing DNA. Well, after having learned all this, I think that we're now able to better understand Jurassic Park and even one day recreate dinosaur DNA ourselves.